The overall goal of this procedure is to identify small molecules which modulate the intracellular levels of the second messenger cyclic di-GMP in the opportunistic pathogen Pseudomonas aeruginosa by means of a high-throughput bioreporter screen.
This method can help uncover small molecules that interfere with Pseudomonas aeruginosa biofilm formation and antibiotic resistance by means of cyclic di-GMP modulation.
The main advantage of this technique is that it is very robust and versatile, allowing the screening of upwards of 3,500 compounds within 48 hours.
The implications of this technique extend towards developing novel therapies which potentially sensitize Pseudomonas aeruginosa to the immune system or to existing antibiotics, while exerting a lesser selective pressure on the bacteria.
Inoculate 5 milliliters of lysogeny broth, or LB medium, with a single colony of P. aeruginosa, 7 degrees Celsius, with shaking at 200 rpm. Then, transfer 2 milliliters of the overnight culture into 200 milliliters of fresh LB medium in a 1-liter flask, and incubate at 37 degrees Celsius with shaking at 200 rpm. Monitor the optical density at 600 nanometers, or OD600, every 30 minutes by taking a 1 milliliter sample from the flask and examining using a spectrophotometer. When the OD600 reaches between 0.3 and 0.5, place 40 milliliters of the culture into a 50 milliliter conical centrifuge tube. Pellet the cells by centrifuging at 8,000 rpm for 10 minutes at 4 degrees Celsius. Discard the supernatant